All right, well, I have the difficult task of giving two back-to-back talks and standing between you and lunch. So I'm going to, thanks to Christian, curtail some of the comments and focus on two important topics. One is drug delivery and one is scaffolds. A number of relationships related to research. You already saw from Christian the differences between limus agents and paclitaxel. Certainly, the paclitaxel compound has been the dominant player in drug delivery technologies in the peripheral arterial circulation. You also saw him allude to this. I just wanted to reiterate that the Katsanos meta-analysis still rears its ugly head periodically, but we've put that to rest. And as he pointed out, this paper in The Lancet about a year ago now definitively shows that there's no mortality hazard. And if you put in context the paclitaxel hazard from the Katsanos paper shown here with the final analysis and all the real-world evidence, there's no mortality risk. However, there are safety concerns purportedly with paclitaxel balloons, specifically related to embolic load and the issue of downstream tissue embolization of paclitaxel and crystalline paclitaxel in particular, and the incidence of no reflow. He showed these data from Stetler and colleagues in The Lancet over a decade ago now, showing that there's no mortality signal seen in coronary disease, but there's a significant patency advantage, if you see here in yellow, to limus agents versus paclitaxel. So now that we're over the scare about toxicity, we really need to think about efficacy. So if this is a liberal sort of interpretation, but if you were to look at the original data of coronary sirolimus and paclitaxel-eluting stents, the freedom from TLRs were substantially better than the bare control. If you look at what we've done so far in the SFA, sirolimus-eluting stents have had abysmal patency rates, and paclitaxel-eluting stents, while we think they're good, they're still not anywhere close to what we were able to achieve in the coronaries. And again, you have to suspend your disbelief a little bit to use these figures. This is from a paper that Elazar Edelman and I submitted many years ago and never got published. But the bottom line is that rapamycin or sirolimus doesn't bind nearly as well as paclitaxel to arterial tissues. And that's really why we've not had successful limus-based agents in the peripheral circulation to date. The main issue, as Christian showed, was that we need to get enough drug in the artery for long enough to get an effect. And if you can't get the drug there in the first place, you never get your effect. We have all these different ways of getting drug, but the balloon technologies that are most commonly used are coated balloons, which have a relatively fast drug release. And as he showed, if you give a drug at a bolus, it rapidly will decay away over time, whereas if you use a stent-based drug release technique, you can tune that release and have it resident for a long time, but you have to have a stent. So how do we achieve stent-like results? Well, this, as Christian showed, was work from our group at CRF, and Juan Granada and his colleagues were able to show in an animal model that if you were to take, for example, in the dash line, Lutonix versus Impact and Ranger in the red and the blue lines, Lutonix actually gets very high drug concentrations at time 0, but it doesn't stick around for long. Whereas the Ranger and Impact, because of the excipient and the drug combination, are able to have higher drug concentrations over time. And this area under the curve will dictate its clinical effect. So how do we do that with sirolimus? Well, there's new technologies. This is the MagicTouch from Concept Medical. The idea was because the limus agents wouldn't bind in a naked form to the arterial wall, the idea was to encapsulate it in a phospholipid bilayer that would allow it to create almost these small microparticles or vesicle-like substances that would diffuse through the arterial wall and then become resident. So as you inflate the balloon, the phospholipid particles are deposited on the adluminal surface and then incorporated into the vessel wall through this carrier. And so the balloon, this is a cartoon of what the balloon will look like. You have millions of these particles spray-coated onto the balloon surface. And you can get tissue concentrations that are very comparable to what we've seen with paclitaxel-eluting technologies. So there's a lot of debate about this, but it is doable. There's a host of other features that I won't go into, but this is what the balloon looks like under scanning electron microscopy. And there's been over 4,000 patients treated globally with a now IDE for both below-the-knee and SFA. So the below-the-knee and SFA trials are both about to start. I'm privileged to be the study chairman, and many good friends, Brian D. Robertus and Eric Sisemski, who you'll see at this meeting, are co-principal investigators for these trials for both the below-the-knee and the SFA protocols. There are slightly different protocols, which I'll go through quickly. This is the steering committee for the below-knee trial. This will be 368 patients randomized 1-to-1 with a 6-month primary and then 12-month powered primary. I'm sorry, this is now a 6-month safety endpoint and then a 12-month primary efficacy endpoint with a 5-year follow-up plan. We'll be following the patients very carefully, and they will get powered secondary endpoints assessed at 6 and then 12 months. And these are the endpoint definitions. This protocol is very reminiscent of that which you saw with LIFE-BTK. That was the first successful trial in the space a year ago. The inclusion criteria will be very similar. Proximal 2/3 of the native infrapopliteal segments, RVDs of 2 to 4, with a maximum lesion lengths of just under 20 centimeters. And they have to have a patent single inflow artery into the foot. For the SFA trial, which is germane to the conversation that we're having, the trial design is a little bit different. It's going to be a 1-to-1 randomization of MagicTouch to all comers approved paclitaxel-coated balloons. I don't think there's equipoise anymore, as you've already heard, in using plain balloon angioplasty as a comparator. This will have a standard 12-month primary endpoint with a duplex primary endpoint of binary restenosis based on a PSVR ratio of 2.4 or greater. This is very analogous to the Sirona trial, which, if you're interested in data in the field, will be presented in part this weekend at CIRSE and then in a month at TCT. Again, it's going to be really an all-comers trial. Lesion lengths will be 15 centimeters or less, and it'll be a 1-to-1 randomization with duplex follow-up. Relatively straightforward trial. This, likewise, will be adjudicated by core labs in all domains and is an IDE trial for this technology and should be starting in the 4th quarter of this year. So if you were to look at the entire suite of clinical trials for the MagicTouch, I appreciate Christian already showing the ecstasy data, which are now complete at 3 years. There's a host of other Asia-Pacific studies that are ongoing with Eddie Choke as the principal investigator. The Sirona trial, as I mentioned, will be presented soon. It will be the first randomized head-to-head trial. That will be a large data set. And then the SURPAD trial is now enrolled. It's 1,200 patients in Switzerland, and we'll hopefully have results in the coming year, perhaps as early as LINK in January. So there's a breathtaking amount of primary randomized data that should be forthcoming. Our hope is that we can show that a sirolimus-coated DCB can achieve similar patency levels compared to paclitaxel DCBs and paclitaxel stents. And that's really where the target will be, hoping for primary patencies of in the 80% range at 1 year's time. So to summarize, paclitaxel-coated devices are safe and effective. We've already proven that exhaustively. Limus-Adins analogs are superior in terms of efficacy in coronary intervention, but have yet to be successful in the peripheral arteries because of differential binding kinetics and pharmacokinetics and dynamics. These newly formulated devices, and I would add to this the Med-Alliance solution family of products, have different sirolimus formulations that will hopefully allow us to get sirolimus-coated products into the circulation here in the United States, and they're actively being studies. And I think we will see several trials in the next couple of years that are going to change how we practice in the FemPop segment. Thanks very much.